Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Matthew Wills, who is Professor of Evolutionary Paleobiology. And today I'm going to be chatting to him about crabs and an evolutionary phenomenon known as carcinisation, which basically means to become more crab-like. Matthew, I know this phenomenon of carcinisation is a form of convergent evolution, but before we get into what that is, let's start with evolutionary trees. What are they and how did people develop them?
Well, evolutionary trees are effectively a family tree of species. So they show us a picture of how species are related to one another. And we infer them from two types of data principally. Historically, people would rely much more on comparative anatomy. So they would look at organisms, they would try to encode aspects of the morphology, and then they would produce a tree from that. So things like, for example, how many pairs of legs does something have, or does it have eyes, and so on. More recently, the last 30 years or so, people have relied much more on sequence data, molecular data, sequences of amino acids and sequences of bases within genes.
And that's basically turned things a little bit on its head, hasn't it? The whole genetic stuff has started to change what we know about the tree of life, hasn't it?
Yeah, hugely. There's a problem in looking at anatomy and morphology, and it's a problem called convergent evolution. This is a phenomenon whereby organisms that are not particularly closely related can evolve in such a way that they look remarkably similar. And they do this because there's a similar set of physical or biological constraints acting on them. So, for example, within mammals, there are two major lineages of mammals. One are the placentals. So we're placental mammals. We give birth to relatively large young. And the other lineage are the marsupials. They give birth to a little tiny neonate about the size of a little jelly bean. And this crawls to the pouch in the mother, and there it suckles and grows for a very long time. The rate of development is much slower. And these two lineages, these two big branches of the mammal evolutionary tree, diverge back in the Cretaceous, or possibly even back further into the Jurassic. And so they're evolving in different regions, in different continents to a great extent. And they're starting from a very sort of modest ground plan, something a little bit like a mouse or a rat. And they're diverging and they're diversifying morphologically in different places and doing so independently. And the remarkable thing is how similar certain species of marsupial look to certain species of placental mammal. A best known example is probably the thylacine, or the Tasmanian tiger or Tasmanian wolf. But the remarkable thing about the thylacine is it looked almost identical, particularly if you looked at the skull, to a dog or to a wolf. The convergence in the morphology is so striking. The shape of the teeth, the shape of the skull, just absolutely stunning. And this has happened through 100 million years or more of independent evolution in these two lineages. So it really is utterly remarkable. But it doesn't stop there. There are marsupial mice that look very similar to the placental mice. There are marsupial moles that look almost identical to the placental moles. Flying squirrels, they've arisen in both lineages, honey gliders and so on. These are just remarkable instances of convergent evolution. It's almost like the two lineages have invented the same things because there are similar ecological and evolutionary pressures on both groups.
And this is kind of converse to what people often think of with evolution. So like Darwin's finches, right? So he goes to the Galapagos Islands and he sees these various types of finches. You know, some have got really long pointed beaks, a bit like tweezers that are great for kind of picking insects off of leaves and branches. And then you've got these ones with the broader beaks for cracking nuts and seeds. So the idea is that they originally arrived on the islands as one species and that over time they've evolved these little differences that allow them to fill specific sort of ecological niches. So this this is divergent evolution, coming from a common ancestor evolving into sort of different types, like twigs on the branch of a tree. Convergent evolution is actually when the common ancestor is much further back, but they've basically evolved to look awfully similar.
Well, that's a really good contrast, because within the placentals and within the marsupials, yes, they are diverging, and they're exploring the possibilities of what you can do as a mammal. But because they're both doing that independently, they're both coming up with the same kind of solutions to similar sets of problems, and that's the convergent bit. So it's one from one side of the tree and one from another side of the tree. So there's a marsupial and there's a placental, each coming up with the same sort of solution.
And it's not just body type as well, isn't it? It can be like little characteristics. So you've got whales and bats have both separately come up with using echolocation.
Oh, this is an amazing example because it's mediated by convergence in the molecules in a gene called a prestin gene. And this has similar amino acid substitutions in both bats and whales. Now, you can't get much more different ecologically, really, than a bat and a whale. I mean, morphologically as well. You can't really imagine two mammals that look more different. But because they are both using high-frequency sound in order to echolocate and in order to navigate the world, they've needed to increase that high-frequency responsiveness in the cochlea. And it's the same gene that's mutated in a very similar way in those two lineages to enable them to do that. And that's really quite mind-blowing.
So this is the thing I find really fascinating, because evolution is not directive. It's not like it's working in a particular direction. It knows where it wants to go. It's little tiny mutations that will happen in genes spontaneously. And for whatever reason, a little mutation might happen that might go, oh, well, okay, so that helps this animal survive that little bit better to go on to have children that will also carry that little mutation. And then another mutation happens. And what's so fascinating with the kind of the whales and the bats is that's happened completely separately, just because it confers that little advantage. So it's evolved on two separate tree branches, essentially.
So evolution typically takes small steps, and for something to be advantageous, it has to be advantageous now. So evolution can't look forward and plan, or it can't make a change now because it will make something else that's good possible down the line. There's no foresight, there's no teleology. But what does happen is certain changes probably make certain other types of changes more likely. And they probably close down other options as lineages evolve. There's one particular reason for this, and that's something called pleiotropy. And that's the fact that organisms aren't constructed in the way that an engineer might construct them. Many structures and genes have several functions, and those functions often aren't very obviously related. So you can make a change that has a beneficial effect, but it also, unfortunately, has a deleterious effect. So the best example I can think of is the conformation of neck vertebrae in mammals. Now, you and I have 7 vertebrae, 7 bones in the neck, but so does a giraffe, and giraffes have enormously long necks. Now, mammals have got painted into an evolutionary corner here. They are stuck with 7 neck vertebrae, not because that's biomechanically the best number to have. If you've got a very long neck, you can imagine in other groups like swans, for example, they're birds, they're not mammals, they have 20-odd vertebrae in the neck. Giraffes are stuck with 7 because if you fiddle around with the genes that change the number of vertebrae in the neck, there are other bad things that happen to you. You produce lots of tumours, you develop very wonky looking ribs, there is fibrous tissue between the ribs and so on. And so there are these deleterious consequences because of this phenomenon of pleiotropy. Things don't just do one job, they often do a whole bunch of jobs. So it becomes more difficult to make radical changes. And that's why body plans can kind of become entrenched or canalised through time.
So we're going to talk about carcinization, which is basically meaning to become more crab-like. So this is convergent evolution. So what makes a crab a crab?
So crabs are within a larger group of crustaceans called the decapoda. And decapoda means that they have 10 legs. Now, the decapods include animals like prawns and shrimps, and also things like lobsters. And those animals have a particular morphology. They've got a cephalothorax, which is a fusion of the head and the thorax. Sticking out behind that, they have an enormously large and muscular abdomen. Now, this allows them to move around in a number of different ways. So they have these 5 pairs of walking legs on the cephalothorax. Very often, the first pair of those will be modified as very large claws or pincers, and that leaves them 4 pairs of usually rather smaller legs behind that they're able to walk on. But they tend to walk front to back. They tend to walk in the direction that they're looking. Behind then the cephalothorax is this great big muscular abdomen, and underneath that they have little appendages that are a little bit like oars, and they're called pleopods. And particularly in the smaller shrimp and so forth, they can beat these through the water and they can swim. So they can walk with their thoracic limbs, they can swim with their pleopods, and they've also got another amazing trick up their sleeve. They can contract all of the muscle in the abdomen. And this is a big chunk of muscle. It's the bit that you eat when you eat a prawn and most of what you eat when you eat a lobster. But when they contract this, they've got a fan of appendages right at the back of the abdomen, and that splays out like a huge, great big fan. And they snap the abdomen down underneath the body and they shoot off backwards at very high speed. So it's an amazing way of escaping predators and of moving really fast. So they're really clever. They've got 3 ways they can move around. Now, we like eating shrimp and lots of other things do as well. So that's a really big meal for a predator and makes them sort of quite vulnerable, quite attractive in that respect. So what crabs have done away with most of that great big chunk of muscle in the abdomen. They've folded the abdomen down underneath the body, back up under the cephalothorax. They've also tended to make the carapace much broader and much flatter so that they can slip between crevices in rocks and so on. This also makes their centre of gravity much lower, so they're more stable. They're more able to hunker down and to sort of grab hold of the substrate or grab hold of rocks, and that makes them much more defensive. And the limbs, they're sort of jointed in such a way that now it's much easier for the crab to move sideways than it is to move front to back. And there's more to this than you might think, because if you're trying to catch a crab, you've probably realised that it will do 2 things. Firstly, it can run in either direction. It can escape to the left or to the right, and you don't know which way it's going to go, and it's the same for a predator. The other thing is, because it doesn't have this great big long abdomen sticking out the back, it can rear up, it can sort of push itself forward, it can raise its pincers, and it can get at you very easily. So it's an extremely manoeuvrable and very agile tank, and it can literally run circles around things that it doesn't like the look of. And so that's the process of carcinisation. Comes with 1 or 2 other things. It comes with fusion of the plates on the underside of the animal as well. And there's another advantage actually. So lobsters and shrimps, when they are carrying an egg mass, it's carried by the pleopods, but it's still quite vulnerable. You can see this mass of eggs sort of underneath the abdomen. Whereas crabs, because they've tucked the abdomen underneath the cephalothorax, they're protecting it. So it's kind of armoured. They armoured the egg mass as well.
So these crab-like things, they've evolved 5 separate times?
So there is a group of carcinized decapods called the Brachyura, and these are the true crabs. And these originate once, and the process of carcinization happens in those true crabs. So the edible crab, for example, the swimming crab, the green shore crab, ones that we're probably most familiar with and most often tend to eat. at least in the UK, those originate once. Now, there is a group of brachyurans that's gone back the other way. So these things can sometimes go in reverse. Those are the Rhaninidae, or the frog crabs. They've started to untuck the abdomen, and they've gone from being wider than they are long back to being longer than they are wide. So they're actually sort of working in reverse. But you asked me the question about separate origins, and the group that sits next to the Brachyura on the phylogenetic tree are the Anomura, sometimes called the false crabs. So these include things like squat lobsters and hermit crabs. And hermit crabs, you probably know, have a soft abdomen, and it's coiled up a little bit like a snail. And that's because hermit crabs use the shells of mollusks, once they've been discarded, to armour their abdomen. And when they need to grow, they find a bigger shell. But some of the Anomura have gone the whole way, and they've become carcinized. So they've come to look very, very similar to the true crabs, to the Brachyura. So there's a group called the porcelain crabs, and they've done it very efficiently. The king crabs, which are a hugely important species for fisheries, and huge numbers of king crabs are eaten, those are a separate carcinization event from the Brachyura. And indeed, there's a group called it's only a single genus called the Lamicidae, the hairy stone crab from Australia. And again, that's become carcinized separately, all on its own. So yes, there are multiple instances of becoming crab-like. And again, that's convergent evolution. But it's something that can be lost again. You've got groups that do away with it. And it's down to what's contingently valuable to the organism or to the lineage at the time. And the reason pleiotropy becomes important is if you get locked into a particular body plan and you can't then put things into reverse. So although we like to think of evolution as being this open-ended process where anything is possible, there are many cases where we see lineages shutting down options as they evolve. One of the things that's gone through the media a lot is a question, is everything evolving into crabs? And it's only within the decapod crustacea that crabs have evolved. In fact, only within the group, the Miura, that contains the Anomura and Brachyura, where that's happened. There are other arthropods that have evolved things that look a little bit crab-like. So there are some spiders that superficially resemble crabs, and indeed they're adapted to living in flowers, and they have a flattened body plan, and actually they move sideways as well. But this is a very superficial resemblance. So yes, humans are not going to evolve into crabs. We don't have the developmental or genomic toolkit to do that. We don't have an exoskeleton. And because we've kind of committed ourselves to a particular body plan, it's very difficult to imagine that however many hundreds of millions of years you give us, we're never going to evolve in that way.
And I know this isn't the only thing that's evolved more than once. So there's lots of different things that have evolved more than once.
Yes, I mean, convergent evolution is a phenomenon that's been studied across the tree of life. The evolution of wings, for example, flapping flight. There are birds, of course, there are bats, and bats are mammals, so they've evolved flapping flight independently. The pterosaurs, a completely extinct group, the sister group to the dinosaurs, and those evolved flapping flight, again, completely independently. And I guess you could also include insects, because although they're very, very distant from the vertebrates, they've also evolved flapping flight. So it's a similar set of biomechanical solutions to moving through the air. There are only so many ways that you can do that. Eyes is another classic example. So complex eyes like ours, for example, with a lens, a cornea, a retina, those have evolved convergently, famously within the cephalopod mollusks, so within octopuses and squid. There are differences in the way those eyes are innervated, how the retina is wired up, for example. In vertebrates, it's done in a very odd way. The optic nerve comes through the back of the retina and splays out over the photoreceptors, in fact. So we have a blind spot, this part of our visual field where you can't see anything. And mollusks and the cephalopods, the octopus, for example, innervates the eye from the back, so there's not a blind spot, much more efficient way of doing things. So there are differences in detail, but the overall form of the eye is just incredibly similar because it's doing a similar job. And we're separated from octopuses by a good 600 million years of evolution, but not just twice. So complex eyes have evolved perhaps 20, 25 times across the tree of life. So there are all sorts of different types of eye. There are eyes that can form an image. Our eyes can do that. The eyes of insects, compound eyes, can form images. They're very good at detecting movement. The eyes of a fly, for example, much better at seeing things move than we are. We tend to see a bit of a smudge. So lots of groups have evolved these complex eye types. There are simpler eye types. There are eyes that can simply tell the direction that light is coming from. So many flatworms have eyes that can just sense directionality, and right down to simple structures that can just detect if a shadow's been cast over them. But that elaboration, that building on those early forms of eye to produce something which is image forming, yes, that's happened many, many times because of the physics of focusing light, the physics of forming an image. There's only so many ways that you can do it. And that's been reinvented multiple times.
Matthew, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our social media. For more information about the Milner Centre for Evolution, you can visit our website.